ఫయాజ్ అలీ గారు.
హలో
హలో ఫయాజ్ అలీ గారు, గుడ్ మార్నింగ్ అండి.
గుడ్ మార్నింగ్ సార్. సార్ వినిపిస్తుందా సార్?
వినిపిస్తుందండి. అది ఆడిబుల్ అండి. థ్యాంక్ యూ వెరీ మచ్ అండి ఫర్ యాక్సెప్టింగ్ అవర్ ఇన్విటేషన్.
థ్యాంక్ యూ సార్ సార్ ఫర్ సెలెక్టింగ్ మీ సార్.
విత్ ప్లెజర్ అండి. యూ సో మచ్ ఎంతూజియాస్టిక్.
సార్
హౌ ఈజ్ పాముల్ రెడ్డి కదండి?
బాగున్నారు సార్.
ఓకే, గుడ్ అండి.
చెప్పారు సార్ వస్తా ఉంటారు మన దగ్గరికి
అవునండి
అని చెప్పారు సార్.
అలాగే మీ ఈవెంట్ కూడా మంచిగా ప్లాన్ చేద్దామండి.
షూర్ సార్. అంటే సిఎస్ఆర్ గ్రాంట్ కూడా వచ్చింది సార్ మన
చెప్పేది
దగ్గరికి.
అదే చెప్పేదండి. అవును,
అదే
చూశాను మంచిగా.
అదే
వెరీ
సార్.
గుడ్ ఇనిషియేటివ్, మంచిగా ప్లాన్ చేశారు. అంటే కొలాబరేషన్స్ అని చాలా బాగున్నాయండి.
Yes,
వెరీ
sir.
నైస్. అది స్టార్ట్ చేద్దామండి.
షూర్ సార్.
మేడం మనస గారు,
Yes, sir.
యూ కెన్ స్టార్ట్ ద సెషన్.
Yes, sir. Good morning to the esteemed speaker, participants, Principal Sir Dr. A. Lakshman Rao Garu. Myself, Manasa, Assistant Professor, Department of Pharmacology, on behalf of VV Institute of Pharmaceutical Sciences, excited to welcome you all today, day 6 of 1 week online faculty development program on cell therapy, pharmacological insights, and future directions, organized by VV Institute of Pharmaceutical Sciences, Kudlavalleru. I welcome Patron Dr. A. Lakshman Rao, sir, Professor and Principal of VV Institute of Pharmaceutical Sciences. Now, I would request Dr. A. Lakshman Rao, sir, to speak a few words about today's session.
Thank you, madam. Eminent resource person of today's session, senior academician, Dr. Sheikh Fayazan Ali Garu, Associate Professor and Head, Department of Pharmacology, Nirmala College of Pharmacy, Mangalagiri, Andhra Pradesh. Participants from various pharmacy colleges. A great morning to one and all for the 6th and last day of 1 week online faculty development program on cell therapy, pharmacological insights, and future directions, organized by VV Institute of Pharmaceutical Sciences, Gudlavalleru, from 9th to 14th December 2024. In day 6, Dr. Sheikh Fayazan Ali will deliver a lecture on cell therapy, pharmacological insights, and future directions. And just I will give the outline about the today's session. The session provides an overview of the pharmacological principles underlying cell-based therapies and explored their evolving role in modern medicine. The topic discusses the mechanisms by which various cell types, including stem cells, immune cells, and genetically modified cells, exert therapeutic effects in treating a wide range of diseases such as cancer and neurodegenerative disorders and cardiovascular conditions. It highlights the key pharmacological considerations, including the cell engraftment, immune response, and the regulation of cell survival and differentiation. The lecture also delves into the challenges faced by cell therapy, such as safety concerns and manufacturing complexities and scalability also. Finally, it outlines the future directions in the field, emphasizing the integration of advanced technologies like gene editing, personalized medicine, and tissue engineering to enhance the efficacy and accessibility of cell-based treatments. On behalf of the management, principal, staff, and students, let me thank the today's resource person, Dr. Sheikh Fayazan Ali Garu, for sparing his valuable time for the academic benefit of the teaching fraternity members. And thank you, one and all. Now I hand over the session to my colleague, Ms. G. Manasa Garu, Assistant Professor and Convener for Faculty Development Program, to introduce the today's resource person to the participants. Over to Manasa Garu.
Thank you, sir. We are very grateful of your insightful words, sir. Now, it gives me an immense pleasure to introduce today's speaker, Dr. Sheikh Faizan Ali, sir. Is an experienced academic and researcher currently serving as the head of the department and associate professor in the Department of Pharmacy Practice and Pharmacology at Nirmala College of Pharmacy, Atmakuru. He holds a PhD in Pharmaceutical Sciences from Jaipur National University and a Doctor of Pharmacy degree from Acharya Nagarjuna University. Dr. Faizan Ali sir has specialized in various areas of pharmacy practice, pharmacology, and pharmacovigilance. He has earned certifications in postgraduate diploma in computer applications and postgraduate diploma in pharmacovigilance. Further, enhancing his expertise in these fields, with a strong research background, he has published 35 research articles in renowned journals and has been granted a patent. Additionally, he is the author of the textbook of hospital pharmacy. Dr. Faizan Ali sir continues to contribute significantly to the fields of pharmaceutical sciences and education, playing a pivotal role in shaping the next generation of professionals. Now, I would like to hand over the session to our resource person. Sir, please accept our.
Thank you so much, madam. I would like to thank management of VV Institute of Pharmaceutical Sciences and Dr. Lakshman Rao sir for selecting me as a guest speaker for this FDP, sir. So thank you very much, sir. I am starting the session. So today's topic is about cell therapy, pharmacological insights and future direction.
Sir, can you please put it in the slideshow mode?
Yes, sir.
Now it is good, sir. Thank you. Yes, sir. Thank you, sir.
Thank you, sir. So, cell therapy, although cell therapy has different types of branches and it is a broad area of research and therapeutic purpose, there are different types of therapies which are related to the cell and which are usually interlinked and but are has a slight of differences. For example, cell therapy. Cell therapy, there would be administration of cells to the body for a therapeutic purpose. Means they will take the cells and they will cultivate in the lab, and later it will be introduced into the patient's body for the therapeutic purpose. And there is another branch dealing with the same type of cell is regenerative medicine. So regenerative medicine means it replaces or regenerates the human cells or tissues or organs for restoring it to the normal function. For example, some of the diseases which causes damage of the organs or decay of the organ tissues or apoptosis of those cells will be replaced by these cells. So usually these are also a branch of cell therapy which is related uh, to the cell therapy, but here it is not for the therapeutic purpose, but it's a regeneration of the cells and cellular immunotherapy. In this branch, uh, immune cells or other type of cells are used to modulate the post-immune system or direct elimination of the pathogen or tumor. So usually these types of cells they use cells like T cells or CD4 T cells or cells to modulate or modify the immune system of the particular person so that they can eliminate the disease or any other pathogen. For example, tumor cells. These cells generally any disease which is enters into the body is acted by immediate response of immune system. When these immune cells fail or not able to cope up with that disease, then disease will develop. So giving strength to this immune system can eliminate different types of diseases. This is one of the successful theory in many of the treating cancers. So it is also a type of branch. These three, cell therapy, regenerative medicine, and cellular immunotherapy, are usually related to one another, but are different branches and uses a different theories. Origin, cell types, somatic cell therapy. So somatic cell therapy, in these cells have been propagated, expanded, selected, and pharmacologically treated to alter the biological characteristics. In these cells are designed in such a way that they will treat those therapeutic purpose or they will serve up to that patient only. What we have given, whenever the purpose is attained, and it will stop working to that patient only. That gene modified or the cell receptor which have been modified or given to the patient will not be carried to their offsprings. Whereas germline cell modification, it is also called as germline gene editing. It is a process which involves altering of genes of reproductive cells such as egg or sperm, or to change the genetic makeup of an individual of offspring. In somatic cell therapy, we don't alter the eggs or sperm, and the cell will not carry to its offspring the modifications. But in germline cell modification, whatever the modification we have done will be carried to the offspring, and it may continues to the generations. So it when compared to germline cell modification, somatic cell therapy is widely being used because it is limited to the patient only, and germline it causes modifications in offspring. Also, it has very regulatory problems and therapeutic challenges that is going to be faced. So germline somatic editing and germline editing. So we will edit in somatic editing a blood cell or therapies therapeutic purpose. For example, for treatment of leukemia, we will take some of the blood cells, alter those blood cells, and later cultivate it and induce into the patient. And whereas in germline editing, so germ modifications are developed and copied into the new cells like a basal cell or a sperm cell. They will be changing the gene, and this will be later induced into the patient, which causes a modification. This modified gene is also observed in the offspring developed from that patient. Like me, they will carry for generations in the children also, and copy. So edited blood cells, unaffected cells, this gene editings contain only target cell type. It will not affect the other types of cells. Means whatever if it is designed to kill leukemias or WBC cells or cancer cells, then it will kill or it shows its action upon only the cancerous tissues or cancerous cells. Whereas in germline editing, the gene copies is in modification goes into every cell or of the including the egg and the sperm, and it causes significant changes according to the edited gene. So risks. So in risks, any changes, including potential off-target effects, are limited to the individual only. This is not passed for future generations. Somatic gene editing. Whereas in germline editing, the person has children and the gene edited will pass to the future generations. So it faces a lot of challenges and consequences. Somatic cells has been therapies has been researched tested for more than 20 years and has been highly accepted by most of the regulatories like FDA, and have been used at various parts of the globe. Whereas germline editing is a new branch, and here the persons are facing some legal and social considerations to use this germ editing therapy. So when we see the historical background of the cell therapy, early in 1990s, the research has been started, and it came into light in 2000. 2000, the first cell therapy which is approved by FDA to treat some of the cancer cells. In latter, further research has been developed, and significant enhancements takes place, and there are development of mesenchymal stem cells in 2006, and later in 2012, after the 6 years, uh, CAR T cell therapy has been introduced, a new concept, and in today's world, CAR T cell therapy has been widely used for most of the therapeutic purposes. Here in 2020, new guidelines has been established for allogenic therapies, taking into considerations of the regulatory aspects at different parts of the globe, taking safety and efficacy considerations of those patient. So mechanisms, mechanism of action. In generally, first cell activation. So cells recognize the abnormal or diseased tissue. Initially, we take some of the cells either from the patient own body or similar cells from the different animals which are having similar cells of the human, or we will use donor cells. These three types of cells we will take, and then we will cultivate the receptors or gene editing will be done into the cells, and those cells will be allowed to develop. After multiplication, it will be transferred into the patient body. After the transferred into the patient body, these new cells grows, and later it gets activated and recognizes the abnormal tissues or diseased tissues. For example, we are giving cell for cancer treatment, then these cells will go for the cancer cells and do the immune modulation. So immune response for better outcomes like killing of those cancer cells. If these are, and later it will go for tissue repair. Means they will promote healing and regeneration in those damaged tissues. Later, these new cells will release cytokines. So cytokines are the one of the uh, plays a key role in signaling of proteins and coordinate these cells for generation and showing its action. Then cells gets migrated. These therapeutic cells moves to the intervention areas and target their cell interaction. This target cell interaction with other cells and influence their behavior. and then they will induce apoptosis, triggering cell death of harmful diseased conditions. So here apoptotic induction generally takes place for those abnormal cells and those cells which has been grown for our therapeutic purpose. Host cells also, some of the cells which we induced by gene edited or somatic receptor mapping cells, these cells also goes into apoptosis once their um, uh, activity or duty is completed and upon long-term persistence. So therapeutic effects may last beyond the treatment periods after the disease get cured or therapeutic goal is achieved. But for using this therapy, the patient should be have some specific or inclusion criteria to this therapy since it is not yet truly developed. So type of allogenic therapies. So stem cell transplantation, T cell therapy, natural killer cells, or dendritic cells. These are the different types of cells used in allogenic cell therapy for treatment of various diseases and cancers. So, in stem cell therapy, stem cells transplantation takes place. So, these are obtained from the bone marrow or peripheral blood, either from the patient or from the donors. So RIT indications are generally used for blood cancers and administered through IV route, and efficacy is shown very high in treatment of cancer cells by these cells. In T cell therapy, T cells are used generally. Donor lymphocytes are the source of cells for this T cell therapy treatment, and it is used for various types of cancers. And it's also in clinical trials and showing efficacy is also very good. Whereas natural killer cells, these are obtained from the peripheral blood from the patient or from the donor, and is generally used for solid tumors. Same, it is administered through intravenous route, and it is still in developmental stage, preclinical stage. It is being developed, and efficacy is somewhat challenging, showing somewhat efficacy still development further is going on for this usage of NK cells to treat these cancers. And dendritic cell therapy. These dendritic cells are usually obtained from the bone marrow. Usually in nowadays these are used for vaccine development of the cancer, and it is administered to intra-arterial route. It's still in research phase, and efficacy is not yet obtained. Target indications, when to use for what are the diseases that are being used for in Japan. There is a high research in this area for the usage of cells for development of treatment of various diseases. Generally, cancer cells to treat cancer, these cells cell therapy is being used for a diversification of different treatment options. Especially at present, hematological cancers are being treated by usage of this stem cell therapy, and autoimmune diseases to restore the imbalanced immune system to the normal, and to treat genetic disorders. Some therapies are still being designed to treat genetic disorders like sickle cell anemia. By editing the gene, they can achieve normal generation of red blood cells and cardiovascular diseases. So, to treat heart cells, for example, some of the diseases like atherosclerosis, and, some hypercholesteremia, these are the diseases usually coming from their ancestors or parents. So, these are cells can be modulated. These diseases even get started when the child is birth within days, even in infants. Neurological disorders. So some therapies aim to neurodegenerative disorders, but there is limited option, not yet into practice. So for example, they are using to regenerate options like Parkinsonism disease and Alzheimer's. They want to regenerate those neurons and organ transplantation, reducing the rejection risk from advanced therapies, which is one of the crucial steps. And even further research is going on with the help of 3D printing technology to develop a new organ which has less rejection rates, or that organ can be developed by using the same patient cells. And to treat chronic infectious diseases, to enhance immune system and treat the immune related diseases like inflammatory bowel disease or any other chronic infections like HIV patients. Generally, these therapies are used in some of the selected patients, usually in patients, adult patients like age greater than 18 to 70 years. And therapies uh, they have undergone, and many of the therapies fail, they can go for this some gene therapy or cell therapy. And physical health of the student should be uh, overall good health status, so that they can be administered somatic cell therapy. And informed consent should be obtained from the patient to overcome the regulatory issues and risks during the treatment disease state. So patient should have a specific disease state. Means advanced stage is mostly preferable. Initial stages we are not able to control the action of these cells after induction to a limited stage. So that's why we are generally preferring advanced disease stages. And genetic compatibility must evaluate the genetic markers of compatibility with the donor cells and immune status, assess the patient's immune system function and patient response uh, for induction of these therapies. So manufacturing process. In the manufacturing process, cell sourcing. The cells are taken from the healthy donor, or usually from similar. It may be similar animal, or maybe a person, or some of the therapies require the patient cell itself. Then these cells are undertaken into processing. They will isolate, purify the cells using advanced techniques, and then these cells will be allowed for expansion to multiply in a controlled environments, so that number of cells will get increased. Then quality of the cells will be tested and functional regularity, and then cryopreservation will be done, freezing of the cells for long-term stability, and then it will be used. And it is formulated. So what are the necessary changes or necessary receptors that to be allowed on those cells will be administered, and then administered into the patient for the therapeutic benefits through infusion into the patient through IV route or required route of administration. So comparative analysis of analogous therapies with autologous therapies. What are the advantages and disadvantages? When we compare the cost of analogous therapies and the allogenic therapies are generally lower cost and is more affordable and widely used, and is generally used for cancer treatments and genetic disorders. And some of the insurance is not available for these types of therapies. Some companies are not giving insurance for many of patients have health insurances. In production time, for allogenic therapies, it requires short production time, very time taken is very less. While as autologous therapy, it requires longer time and faster treatment results can be obtained, and but urgent needs cannot be obtained. So it can be used in cell replacement therapies. In scalability, highly scalable therapies can be obtained in allogenic therapies, but autologous therapies is limited to individual donors. Advantage is easier of the scale production, and when this is dependent on the donor availability. Generally applied in regenerative medicine. Patient available. So it is widely accessible, and autologous therapy is accessible only for donors in streamlined patients, limited to donor criteria and autoimmune diseases. Healthcare system impact on allogenic therapy. The main strength is it provides the standardized treatment for the diseased patient so that he can overcome the diseased condition, and it can be um, immediate patient who can need immediate cell therapy can be obtained or undergo for allogenic therapy. So it requires a very short time for production, and many of the researchers are going interest in this regenerative medicine. So opportunities are very widespread, and expanding applications of in chronic diseases is significant in this market for usage of allogenic therapies, like and weaknesses. So when we compare the weaknesses, the production cost is little bit high, and all the populations may not afford this, and regulatory challenges are also being faced. When compared to threats, therapies may impact some market share and adaptation, and safety, efficacy, and patient high acceptance is also one of the threats. So this is the SWOT analysis for the allogenic therapy. So when come to the future trends in cell therapy research, there being researches for development of targeted therapies. So allogenic therapy targeting specific disease for precision medicine, and through going engineering cells. Like they want to cultivate the cells which target a specific disease and cure, and later it will get into apoptosis or necrotic mechanism going out of the patient's body. Uh, Specific cell destruction they are targeting. In regulatory advances, still it is in process and challenging condition. So streamline regulation should be uh, processed for allogenic therapies and accelerate its market access so that it can be widely used in many of the patients, and it can be when many patients can be benefited, and enhanced safety. So new technologies are going laying way for safety proper allogenic therapies, reducing the adverse effects. So sometimes they're not able to differentiate or not able to control this cell after inducing it to the patient. Sometimes immune system is responding against these cells and causing some type of rejection reactions, and some uh, adverse drug reactions are also been observed, and adverse events are also observed by using this cell therapy. So there should be uh, enhanced safety. So new engineering techniques are there coming into the evolving into the market for safety of these therapies. Scalability. So these allogenic cell therapies are usually produced in small scale, but there should be an advancement of research, and further it should be developed for large scale production of allogenic therapies, increasing the to meet the global demand, like how we ready-made take some tablets or like that. This also should be available. So the future therapy is generally focused on expanding the treatment options, a wide range of diseases. So most of the this is not these are limited to up to some of the cancer cells, and some degenerative diseases, but most of the diseases can be treated in future by using this cell therapy. This can be obtained by improving the safety and efficacy of the specific cell population, and developing off-the-shelf allogeneic therapies means readily available. At present, we are at, when the patient requires the treatment, we are evolving those type of cells and then later inducing into the patient. But for example, nowadays, if you want a vaccine, when we go to the hospital, their vaccines are readily available. They are directly injecting for any disease. The drugs are available in the hospital, and the pharmacy store we are getting readily. Like that, cell therapies are not available. So off the shelf therapies should be come into the market, so that whenever the patient requires, there should be cells readily available and can be injected into the patient to control those diseases. So this type of technology should be developed for off-the-shelf allogeneic therapies. This can be developed by using of advanced genetic engineer techniques to enhance the cell function and addressing manufacturing challenges to make cell therapies more readily access to the patient, like these storage conditions on manufacturing, increasing the shelf life, increasing the specificity, uh, less rejection reactions, less ADRs, increasing safety profile and efficacy profile of the patient. So, ongoing clinical trials for iPS and ESL based therapies. Usually in Japan, there are trying to generate this cell therapy in treating various number of diseases. So they want to treat by this cells using age-related degeneration. So they want to prevent like retinal pigmentosis, yeah, and limbal stem cell deficiency. They want to treat heart failure, cardiomyocytes can be developed for articular cartilage damage, rheumatoid arthritis, or osteoarthritis. They want to generate cartilaginous tissue for immune cancer cells. They want to use immune cells so can fight with those cancer cells, and to treat thrombocytopenia. They want to develop platelets that can urate and urea cycle disorders. They want to develop hepatocytes, and to overcome the spinal cord injury, they want to generate neural progenitors, and Parkinson's disease. They want to develop dopamine neural progenitors cells. So there are various researches going on in this area cell therapy in Japan to overcome most of the diseases. So when we see the FDA approval and the clinical trials that has been performed in Japan, there are different types of cells uh, has been developed and came into the some of the market with specific guidelines for some of the patients which who meets the inclusion criteria. These they are developed cardiomyocytes for heart failure, anemia, to treat aplastic anemia, advanced heart and neck cancer treatment, to develop cartilage damage and urea cycle disorders and renal pigmentation. These types of cells are being developed. So, which came into the market drug in the name of bosutinib and roprinolol hydrochloride, brochrompatine, and sirolimus for inner ears and mitochondrial and more develop motor neurons. So they have developed, this study shows they have developed nephrons through this cell to overcome kidney disease. If you see the graph, so after the transplantation of the cells, this blood urea nitrogen levels are getting declined. Initially, there is a spike in the blood urea nitrogen levels, means kidney function has been deteriorated. After the cells has been transplanted, then the cells response of the blood urea nitrogen levels is getting decreased. Meanwhile, same in the creatinine also. There is a decline in the serum creatinine levels up to the nearly normal after 12 to 14 days of administration of these stem cells. If we see the cells in this histopathological observations, there is a outcome, positive outcome in response to the cell culture. So they used this cell stock, they cultured and differentiated, purified, and then they cryptopreserved these cells and later shifted into the cell therapy for kidney transplantation diseases. So what are the areas of future development? So broader cancer application. So present these type of cells are generally used widely used for treatment of hematological cancers like blood cancers and like leukemias and lymphomas. So researchers are actively trying their level best to overcome for solid cancers like lung cancer, pancreatic, brain cancer, developing numerous strategies for improving their tumor homing capabilities. So they are trying these T cells, especially for this cancer overcome treatment. They want isolating those cells from the different sources, and they're using technologies and activation of preparing a receptor on those cells and later expanding those cells and introduce into the uh, patient body. So immunomodulatory cell therapies. Cell therapies. So utilizing immune cells like regulatory T cells to treat autoimmune disorders by modulating their immune response and promoting tolerance. So in T cell therapy, tumor or blood samples are generally taken, and T cells are being isolated from those samples, and those are allowed into for multiplication to enhance their function in the lab, later delivered back into the patients. And after entering into the patient's body, these T cells will continue to multiply in the patients, and later, after increasing the population of those newly generated T cells, they will recognize those cancer cells and kill those cancer cells. So T cell therapies. So TIL therapy, TCR engineered T cells, and CAR T cells. Usually nowadays, CAR T cell therapies are widely being evolved throughout the globe. So TIL therapy, tumor sample required generally a surgery is required to obtain tumor tissues, and target antigen generally not need to be identified as tumor cell, like T antigen. This does not require generally genetic engineering technique. It generally likely target those because those are diseased cells. They will be modified on those cells and later induced into the patient. They usually go through many and get a better number and attack those tumor cells. And TCR engineered T cells. So TCR engineered T cells usually uh, tumor antigen is identified in advance, and antigen must be displayed on a cell surface. For this, a T cell target has to be evolved on those surface, and by using genetic engineering, it recognizes the tumor antigen and displayed on the cancer cell, promoting T cell attack. So I received a doubt: how does this T cell recognize cancer cells? So generally, these T cells in TIL therapy, there are those tumor cells. So they will be similar cells will go and attack. And in TCR cells, we are developing an antigen, the antibodies on those surface. Those antibodies will be attached with those antigens there. So like that, they will recognize those cells and attack on the required tissues. And CAR T cell technology. So through blood, we will take these cells, and a tumor antigen must be identified in advance, and antigen must be present on the cancer T cell surface. Later, genetically engineered receptor is developed by using CAR T cell technology with a known antigen, which is genetically engineered and transferred into the patient body for further attack. So stem cell based regenerative medicine. So stem cells, they are being used to regeneration of muscles, blood, bone marrow, heart diseases, neurons. These are being used to stem cells to generate specialized tissue for regeneration different types of conditions. In nowadays, nanotechnology has been playing a very good role in development of these uh, cell-based therapies. For example, stem cells are used for development of cardiomyocytes and tissue repairs, and macrophages for drug delivery system into the brain, and somatic cells for induced direct reprogramming of the neurons. So this can be done by injection of viral or non-viral containing therapeutic environment into the patients. First, to remove cells is removed, then correction of cells is done, and autologous transportation has been done through correction of the cells later induced into the patient. So, gene editing technologies. So generally, uh, CRISPR-Cas9 technology is one of the widely used among all the technologies for a gene editing tool and modifying the gene function. So it is potentially allowing cells to correct the gene. So it's like a reprogramming, finding and replacement of the gene technology from the cell. We will we treated with an enzyme containing complex gene, so guide the molecule and that where the gene is modified, that is removed from the standard synthetically designed gene is later attached by DNA cutting and replacing it with healthy DNA. The defective gene is generally removed and later the cell will develop with the help of the edited gene into a new cell. Later it will be progressed and transfused. In therapeutics, cellular therapeutics, and extravascular vesicles, anti-inflammatory medicine, cellular patches has been developed. With the challenges we are faced in these gene editing technology therapies, is stem cells heterogeneity, some of the adverse effects are being uh, experienced by the patients, and malignant transformation also can occur, and efficacy and feasibility is also a main challenging part, and there are technical limitations to control this multiplication, or how much effectively the cells get multiplied, and what might be the life of those cells, and what the capacity of regeneration and reproducibility, and main challenging factor, rejection reactions. So advancements in the pressing manipulation of enhanced cells, and to deliver some of the dose specification should be developed. How much cells we have to induce for a particular stage of the disease, and allogenicity of autologous disease based upon which type of cells can get in. Tissue engineering construction, and in vivo delivery of the cells to improve recognition of donor organs. And clinical trials are being widely developed for some of the acute and chronic diseases, and COVID-19 work in COVID-19 also. This type of research has been taken in the USA and London, and to treat autoimmune disorders, and to treat wound healing, and improve transplant tolerance for organs or tissue regeneration or repair. Main goal is of these therapies is to promote endogenous regeneration, reduce the inflammation of chronic inflammation diseases, and delay in the progression of the disease. If not possible for some of the cancers, if they cannot cure, they just delay in the progression, like how there is anti-cancer medication some of the times, and provide personalized medicine depending upon for this particular patient, and reduce the elimination of immunosuppressive agents, because these immunosuppressive agents, even though they are essential for some of the diseases, there is a risk of developing another type of infection that the weak immune system is a weakened. So, like the replacement of immunosuppressive agents by these cells, and avoid whole organ transplant. So generally, um, this is not only this cells therapy is not only used in our healthcare system, but different types of biofuels are being developed. System crops development, new industrial products are development. Even Japan is trying to develop some of the uh, plants or crops which are free from diseases. Nowadays, uh, many plants you can see they are genetically modified and are usually available in the market. Many fruits and vegetables which does not grow to our condition, they are genetically modified and prepared in such a way they are able to grow in our conditions like that. So challenges they are being faced is safety, ethical concerns, and regulatory challenges. So off the shells, in the area of off the shells allogenic therapies, developing a donor derived cell products So that can be usually readily available for the multiple types of patients, like how that medicines and vaccines are available in the market, like that readily available cells for patient specific condition, and manufacturing and huge manufacturing, large scale manufacturing should be required in reducing the timeline. For example, healthy donors should be taken. Cells are extracted, different types of cells, natural killer cells or T cells, and then we can insert a readily made CAR gene insertion and gene editing is done. New cells are developed. Those cells should be storage, and if the patient comes, then we have to give this administer those cells. Like how we are presently giving prescribing medicines, like that, an off the shelf cell technology cells should be developed and stored so that they can readily administer without delaying the time that the patient has to come. Later, we have to find a donor or find these cells. Later, we should gene editing should be done. Later, should be multiplied to reduce gene types should be ready-madely available. So for this enhanced CAR T cell designs can be used. So thymic antigen receptor CAR construction improves specificity, potency, and persistence of these T cells, addressing different types of side effects like cytokine release syndrome, like that. So different types of cells can be used. For example, tumor cells can be used, and healthy cells can be used, and cytokine release syndrome. Different types of cells can be used, like interleukin 6, interleukin 8, gamma, interferons, like that. And for T cell trafficking and infiltration, vasculature, there are some of the limitations up to, and for fibroblast and dendritic cells. So for CAR T cell therapy, so patient T cells can be removed or donor T cells can be removed that are developed in the lab and gene edited using CAR T. That CAR T protein is developed, and these are infused into the patient so that they can kill the antigen antibody binding. So FDA approved CAR T cell therapies like MRI, CD9 cells or antigen is targeted, used for treatment of lymphoblastic leukemia, lymphoma, these types of cells, usually administered to various cancer patients available in the market. So some combination therapies, development of combination therapy with the usage of cell therapy, and they can use similarly along with radiation therapy, chemotherapy, immunocheckpoint that can be developed for synergistic effect for treatment of cancer cells, and the usage of advanced cell engineering, so biomaterials and 3D technology for used for tissue generation, tissue printing. So different types of particles can be used, like gold nanoparticle can be used for delivery of some of the drugs, and carbon nanotube particle for development of tissue including bioprinting and like that, development of organs, and iPSC induced pluripotent stem cells for development of some of the tissues, or muscle regeneration or nerves regeneration, like that, and mesenchymal stem cells for growth of some enhance the growth factors. So these are challenges to overcome the cell therapy development. So manufacturing scalability. So generally, to develop strategies which are cost effective and efficient method to produce large quantities in high scale for industrial purpose for development. Present technologies is very high cost to, and the small scale development is available. And immune rejection. So immune responses after transplantation, we're using allogenic therapies, and delivery methods. So different strategies for developing this appropriate cells into the body, and clinical trial design, particular design for development of clinical trial protocols to enhance the safety and efficacy. Summary. So future directions. So therapeutic potential, allogenic therapies can address wide range of diseases effectively. Regulatory considerations should be developed. Market growth, allogenic market cell therapy is being enhanced and a number of investments has been increased. So collaborative research should be developed. Partnership with the scale holders like company and academics should be both the two collaborate research and advances in manufacturing to reduce cost and increase accessibility, focusing on the needs of the treatments and acceptance of the patient. So findings. So this sector is growing very widely throughout the world, and there is a increased scope of funding in clinical research activity. As the at the end of 2023, 76 cell and gene therapies has been launched globally, which is double the number which had last in 2013. And patients who are eligible for gene therapies, a complex and numerous barriers exist through this process. So this can be overcome, and number of $1 billion can be scaled up to 5.9 billion has been used in 2020, which is 38% more when compared to previous year. And globally, there is 1.6 trillion money has been invested in the research of these stem cell therapies. So uncertainty exists in future trajectories and sustainability of gene cell therapy. So these are the indicators which are being wisely used, and different scenarios for uh, venture capital amount will be used for development of this area. And over the last 5 years, globally, 3,285 trials has been developed to evaluate these types, and including only gene therapy patient across sponsored in 631 2023, and non-industrial trials in academic institutions and non-profit NGOs have been increased to 36% in compared to previous year, and while industry trials also accounted for 64%. So you can see, especially CAR T cell therapy is growing very widely, and latter occupies the stem cell therapy. And global cumulative cell and gene therapy launches different, as you can see. Gene therapies have been evolving in last previous 10 to 15 years, and cumulative research has been increased to determine data. So, in conclusion, cell therapy is one of the evolving and exciting promising clinical potential, which is going to show miracles. In order to allow commercial use of these therapies, so manufacturing and cultivation process is still should be developed and dramatically improved to allow clinical to meet the clinical demand globally. Further, some of the cyclic GMP standards for second signaling in process of controlling parameters are constantly monitored and analyzed, and some of the living cells can be used in these therapies, and their effect on by these therapies should be monitored, and different types of different cultural requirements raising from the suspension of adherence as very phenotypes and characteristics, and no single culture platform can fill all types of cell. So different requirements, different new cells to be developed in a controlled and monitored environment by using such as equipment such as bioreactors, and choosing a correct type of cell to attain therapeutic purposes, which is not an easy task. So there should be we should take into account the batch size, cell quantities, and available technologies, and further research should be more developed in the usage of tissue kinetics and limitations of this cell therapy. And downstream post culture processing we should be corrected and precisely defined. So this stage of usually shortest stage, but is one of the most critical step in this process. And contamination during this stage, failure can be. So stress induced to the cells during the critical stage will defect in the final product. So therefore, some technology should be developed and still should be processed to overcome this stress induced cells. So experiments and capabilities of step cell growing, and although there are some of the obstacles practically have to be overcome. So regenerative medicine and transplant technology is one of the uh, evolving parameters, and some untreatable neurodegenerative disorders can be treated by using these stem cell therapy in future. And induction of pluripotency enables patients' own cells tissue bank should be are growing globally. These are times we can say many of their centers are using children's umbilical cord stems for this stem cells in the future for regenerative medicine that can be used. It's an evolving branch with stem cell therapy. All the regenerative benefits are able to attain better and provide human life, uh, prolong human life in every time in the history. Any queries? Thank you. Thank you, sir.
Thank you, sir. Thank you for your sharing insights on the topic. Thank you for
Thank
sharing
you,
your
ma'am.
expertise, sir.
Thank you
Now
so
the
much, ma'am.
yes, sir. Thank you very much, sir. Now the floor is open for discussion. If the participants have any queries, can you ask? If the participants have any queries, you can ask. Okay, sir.
Thank
Thank
you,
you, sir.
sir.
Thank you
Thank you,
very
madam.
much, sir. Thank you for your valuable time, sir.
Thank you. Thank you so
And,
much, ma'am.
and
Thank you so much, sir. Okay, sir.
sir, 1 minute,
Shall I?
sir. We will give an appreciation certificate, sir. I will share it.
Okay, sure, ma'am.
Sir, it will be visible, sir.
Yes, madam.
Yes, sir.
Thank you.
Thank you, sir. Thank you very much, sir.
Thank you so much. Okay.
Yes, sir. And I would like to thank all the resource persons, participants from various colleges, and our principals, sir, for giving this opportunity and make this program a very grand success. And let's end this session with national anthem. Yes, sir. Thank you, sir. Thank you very much, sir.
Thank you.
And all the participants, I'll provide a feedback link on chat. Please give within 10 minutes. Thank you.